Hi everyone, I'm Soumya and I'm working with Ishaal and we're showing textbook pages 115 through 120, which are about the value of animal models within psychological research. So within psychological research, animals serve as a representation of evolution. So as humans share characteristics with the animals that are most prevalent within psychological research, and these animals include rats, pigeons, chimpanzees, cats, rabbits, etc. So, within this research, there's something called comparative psychology, where investigators can choose to put their attention into a specific species or explain how that species would compare to humans and the characteristics of humans. Additionally, there's a theory called the triune brain, which states that the brain is divided into three regions: the reptilian complex, the limbic system, and the neocortex. This was developed by Paul D. MacLean and is one of the early stages of a model. But it has been proven to be inaccurate. Initially, it was thought that this structure could represent animals as well as humans, providing evidence of evolution. However, we have proved that our brains have evolved in a more intricate way and they have not built off of one structure to the next. Animal models are used in order to test an experimental hypothesis regarding human actions using animal research. These non-human subjects are used due to their ability to mock results that could occur on humans, for example, displaying how a disease will progress. So there are four types within these animal models of manipulation. So the first one being genetic manipulation, which shows a genetic modification where the DNA of an organism is altered, and this is done using technology within a laboratory. The second one is invasive manipulation on the nervous system when parts of the brain are affected due to lesions, electrodes, or simply just due to removing a part of the brain. Invasive manipulation on other body parts is where parts of the body are affected by different substances, and in the process, the body parts are damaged. And then lastly, there's behavioral and environmental manipulation, which is the manipulation with the intent of achieving a target behavior. So, for example, if you're preparing healthy food at the start of the week, it's an encouragement to stick to the goal of eating healthy throughout that rest of the week. Comparing psychological functions. So, scientists argued that by solely comparing the differences of the brain among species, an accurate representation of the similarities between animals and humans is not going to be shown. So, in order to compare the two, a man named Premack voiced that we needed to focus on the differences between humans and animals. So, some examples of those are how we teach our young. Animals teach their kids a completely different way. For example, if a cat was to bring a dead mouse to their young, humans don't exactly teach their kids to do that. Humans teach their kids how to eat, but a cat would bring the food to them and expect them to learn how to eat on their own. Additionally, we have short-term memory, so our memory can be relatively similar to that of a chimpanzee's. However, they will still differ because chimpanzees will see the word 11 as not the word 11, the number 11 as two numbers, individual numbers, rather than chunking them together like a human can. A human would view the number 11 as one number, whereas a chimpanzee would view it as two. So the advantages and disadvantages of animal models are that the advantages are that human and animals share similarities within the structure and genetics of their brain, and the use of animal models has been proven beneficial as it resulted in the discoveries of numerous life-saving treatments. Additionally, the lifespan of a lab mouse creates idealistic conditions for researchers as they can easily observe the mouse's entire life. This is due to a mouse only living 2 to 3 years, whereas a human test subject would live and potentially outlive a researcher. Animal research allows for more control over confounding variables, and this increases the internal validity of the research. And animal models are useful due to their inexpensiveness and easy accessibility. But there are disadvantages, though, as animals and humans will never be exactly identical. Animals and humans may conflict psychologically, and treatments that are tested on mice typically need to be done on a larger species before a trial is done on humans. There is also an environmental factor to take in, as laboratories could put stress on a subject. Results shown as negative in humans could be positive within animals, and then vice versa.
There's four main examples of animal research, the first being brain and behavior or localization, and then brain and behavior and neuroplasticity, and hormones and behavior and genetics and behavior for epigenetics. Brain and behavior in terms of localization An experiment performed by Lashley involved rats, and when she removed different parts of the rat's cortex to see its effects on the rat's memory of a maze that it was put through. And her results showed that it was not the part of the cortex that affected the memory, but it was how much of the cortex that she removed that affected their ability to memorize the maze. And this experiment represented localization as it refuted the idea in relation to memorization, and because it was not a specific location in the brain that affected their memory, and that was insightful into how humans functions in correlation to other human research. But the experiment can definitely not be replicated on human subjects because you can't remove a part of the human cortex. Brain and behavior in terms of neuroplasticity. In Menench's study, he aimed to make cortical representations of adult owl monkey hands, and this study showed that there was special brain matter responsible for moving one finger, which linked movement to the other fingers. This experiment gave meaningful insight to the fact that the brain can re-specialize and adapt based on injuries or structural damage on parts of the body. And this could not be replicated on humans, as scanning of the brains itself is extremely harmful. And the taking pictures of the brain causes a lot of radiation, which is harmful for humans. And that's why it's better to get an insight from an animal's point of view on the case. Hormones and behavior. Romero's experiment focused on the effects of oxytocin in relationship to social bonds in dogs in a non-reproductive way. Dogs and their owners were placed in a room. Some dogs got sprayed with oxytocin while others did not. And results showed that these dogs that were sprayed showed higher rates of affection towards their owners. This experiment could not have been performed on humans because the hormone oxytocin does not affect us in the way that it affects animals, because you would need a much higher dosage. And this experiment helps aid us in the effects of this hormone in relation to effectivity. Genetics and behavior and epigenetics. Another experiment was conducted by Weaver, and he tested whether the type of nurturing a rat got from their mother in their early life affected their brain's processes to stress later in life. This was research that found that the less nurturing in early life resulted in the rat suppression of their glucocorticoid receptor, but the genetic sequence itself did not change. This experiment showed us the effects on the gene suppression in relationship behavior, and this could not be replicated in humans as there would not be participants willing to openly give less nurture to their babies at a young age. And with animal testing, there comes a lot of ethical concerns that you also have to take into consideration before experimenting on animals. The first one was all experiments have to be justified with clear scientific purpose. So there has to be meaning behind the experiment that you're putting the animal through. And in non-human cases, the species chosen should be the best for the experiment, and pain in regards to humans should be taken into consideration. So if you think that putting a needle within a human is harmful, then you probably shouldn't be doing it to an animal. And all animal studies should be submitted to the ethics committee before beginning. The ethics committee is the one who reviews like with whether an experiment was ethical or not using animals. And administrator of experiments should be familiar with typical behavior of test animals to detect any illness or discomfort. So the person should know if you're experimenting on rats, they should observe rats' behavior prior to the experiment so they know if one rat is acting a little bit different than normal. Treatment of these animals must be done humanely in humane conditions. They have to be treated like a regular animal. They can't just be subjected to neglection. An experimenter should minimize discomfort for these test animals, and should tests be painful stimuli to humans before administering on animals. Once again, testing on humans before they do any harmful treatment to animals. And if a test animal is showing signs of illness or disease not fit for the experiment, then they can be euthanized, because the rat is no longer used for their experiment, and there's no longer a purpose for the rat or any other animal in these terms, because they cannot be released back to the wild. And so any test animal in a laboratory cannot be released back into the wilderness because of contamination and other things like that.